Well, while this is loading up, let me just say hello. Thank you very much for the organizers for asking me to speak and giving me the chance to share some comments about the research programs of the International FOXP1 Foundation. My name is Samit Dasgupta. I'm a professor of mathematics at Duke University in my spare time. And the main thing that I do is I, work on trying to lead the research direction of the FOXP1 Foundation. Okay. All right, so let me just start with a picture of our kids because this is all about our kids. I had a chance to meet a lot of your kids last night, and I look forward to meeting more today. And my son Ishan is in the upper left corner. He couldn't come. I would try to take him on this flight. Okay, it's working. Yeah, it's not working. Nope. Yep, it worked just for a second ago, and now it's not. Let me just say 3 apologies. I have one is there'll be a lot of repeat for those of you who came yesterday to my talk, and I apologize for that. Also, I'm going to probably go a few minutes over because I had some questions last night about explaining an ASO upregulation, so I added some slides about that. And what was the third thing I was going to apologize for? I've forgotten. Oh yeah, the third thing is that I'm going to go kind of fast because I have a lot to say in a little bit of time. All right. There we go. Okay. So we have a global census that we run. Oops, now it's going without me pushing the button. All right. Ah, oh, you're doing it. Okay, sorry. All right. Let me do it. Okay, okay. So we have a global census that we run, and I want to thank Gail Clark from our foundation again for running this for us. It's a lot of work to figure out how many members we have worldwide, and you can see the growth over time. 652 is a lot bigger than what the number was 2 years ago. This number is made mainly from our foundation signups. So if you are not signed up for the foundation, please do so. In certain countries, we use the local numbers, so please connect with your ambassadors in those countries to get counted from your nation because they have more signups than we do at the foundation. So to get a more accurate global number, we use those numbers. So those countries, I think, are France, Germany, Netherlands, UK, Australia, and Canada. So if you're in one of those countries, please reach out. What's that? And Spain, thank you, yes. Reach out, if you're in Spain, reach out to Lorenza. So we'll make sure you connect to your local ambassadors in those countries. And it's really important, it's not working again. It's really important to get this number as high as we can because that will incentivize researchers and drug companies to work with us. The larger we can make this number, we really want to get over 1,000 as quickly as possible. That's kind of a really important line to cross to incentivize any company to work with us. Yeah, this is not working. Can I just like push the computer? Okay, so I'll just point to you and you do it. Okay. All right, we know that those numbers are still, even though they've grown, they're still a vast underestimate of our community. So GeneDx is just one testing company within the United States, and they told us in May that we had nearly 500 people tested with FOXP1 mutation just by them, and over half of those were with the pathogenic or likely pathogenic variant. So this number of 274, that's more than the number we have signed up for the foundation, 201. There's another testing company that told us they had 45. So if you add those together, that's more than 300 people with a pathogenic FOXP1 mutation just in the United States from just 2 companies. And in the foundation, we only have 200 signed up from the US. So we're vastly underestimating or missing our population. And that's just the diagnosed population. Most kids with FOXP1, probably as a factor of 10, are underdiagnosed at least. So there are probably thousands, maybe 10,000 patients with individuals with FOXP1. We just don't know. We're stuck at 652 right now. Can I point? There we go. Okay, so what is the goal of the research pillar of the FOXP1 Foundation? We do a bunch of things, and probably the most important is the direct funding of research projects. We also host research conferences every even number year, starting from last year, so kind of alternating with the family conferences and the odd numbered years. And there will be one in UCLA next summer. Okay, next slide. Everything we do on research is guided by our excellent scientific advisory board with leading researchers in neurodevelopmental disease and FOXP1 in particular. We have a couple members here today, Dr. Cheryl Fife-Meritich, who spoke earlier on gene therapy, and Dr. Gudrun Rappold, who's somewhere in the audience. Okay, oops, went backwards. Okay, forward, please. Nope. Oh, it's working now. All right, should I try or not? Okay, it's working. So let me do it now. Okay. All right. So we have a patient data collection site called RareX, and I really hope everyone signs up. And even more important than that, we have 256 people that have signed up for this, but only roughly 60 that have submitted their genetics report. And your sign up and your data that you provide becomes much, much more valuable if you submit your genetics report. So let me just please ask everyone here, if you signed up for RareX, please go in and check that you've uploaded your genetics report, because it doesn't help as much if you don't have that diagnosis. A lot of times parents ask me, like, here's my kid's mutation. What does this mean about their outcomes? We can't answer that question unless we can match up parents reporting symptoms with their mutations and then studying that. And so if we don't have the mutations uploaded, then we can't study that. So please upload your mutations. Another data collection site that we've recently joined with is Citizen. So right now, this is mainly operating in the US because of how Citizen works. They get medical records directly from doctors, from clinicians, and then they extract data from that and systematize it so that everything is encoded the same way. So if one doctor sees a child and says this child has low body tone, and another doctor writes that they have hypotonia, well, that's the same thing, just in different language. Citizen will line that up and make a systematic database that researchers can download and see. And all that data is de-identified. So if you donate your child's medical records, when it gets translated into sort of research data that's sent to researchers, the child's identity is taken out. Right now, it's only done in the US because they use the HIPAA laws to obtain the medical records. You don't have to upload anything for Citizen. It's very easy. You sign up and you just enter what clinics your child has been seen at. Oh my God, I have like 15 minutes left. Okay, 5 minutes. Okay, so what do we do at the FOXP1 Foundation? There are three, I would say everything is under de-risking. We want to create an incentives for companies to come in and find treatments for our kids. But we're also specifically studying treatments. Okay, so there are 4 categories of treatments. Small molecules, ASO upregulation, which I'm going to speak about, gene replacement, which we heard about earlier today from Cheryl, and gene editing, which is a little bit more speculative right now for central nervous system conditions. So I won't say much about that. And we're also building up the clinical apparatus, the clinical trial readiness, and making sure we're ready for the regulatory pathway when we want to get to trials. So I won't mention all of these projects. So these are projects that we funded in 2024, a total of $274,000. I'll just mention this project of Dr. Rappold and Dr. Folek. I would say this is the medicine or the treatment that I'm most excited about. Dr. Rappold presented yesterday on an excellent mouse study that showed that treatment of mice that had FOXP1 knocked out was effective with this drug. This is not a drug that's available on the market. This is a non-approved drug. So it's something that we could develop, and I think that, and so we're funding now is to test this on human cells. So some cellular models of the striatum, a part of the brain, called organoids, and just sort of test this drug on those organoids. And so we're funding this project, and we expect to have, good results within 6 months to a year. And at that point, we can move towards drug development and actually getting to trials with our kids. So we're talking about 6 months to a year from now in the most optimistic scenario. And another project I'd like to mention is the development of a CGI scale. This is a particular clinically validated endpoint that the FDA has approved of for other conditions. You need to get it tailored to your particular condition to use it. We need to have these endpoints. These are things that the FDA says, hey, if you meet these markers in seeing improvement for your kids during the trial, that means that the drug is working. And so you need to develop all that apparatus. You can't just show up with the drug to the FDA and say, approve this. You have to show them that you know how to measure that the drug is working. So we have to invest in all of these things. Last year in 2024, we put out an RFP, that's a request for proposals, and we are very excited to get proposals from leading researchers throughout the world for research on FOXP1. We funded 3 projects, which will cost about $300,000 over 2 years. And I won't talk about all of them because I don't have time. Very quickly, the top project, the goal is to demonstrate that FOXP1 can be treated and explore the therapeutic window. So try to demonstrate that adults can be treated. It's a mouse project, and they'll basically try to show that adult mice can be improved. And so that would give us a window of hope that I know a lot of us have adult children with FOXP1, and we want to know that these therapies are going to help them too, and we want to establish that. We're running something very important called a natural history study with Dr. Wilson and Dr. Lord at UCLA. They are going to follow 40 FOXP1 patients over 2 years with multiple measurement points to create a baseline of understanding the development of FOXP1 children. Okay. So this is all for a purpose of getting towards clinical trial readiness. We can use that data in clinical trials. All right, now as the foundation has matured after the, in the past couple of years, we're now conducting research directly with contract research organizations and funding academic labs directly rather than like receiving proposals, but sort of directing research. And so I'm going to speak for a little while about ASO upregulation. We're making a major investment into developing ASO upregulation, which is a possible therapy for these kinds of diseases, and I'll talk about what that is in a moment. And we heard a lot about gene therapy this morning. We want to fund a proposal, so if you were paying very close attention, there's a part of the AAV, that what goes inside the AAV is called an enhancer and promoter. That's a really important part of the transgene that you need to design to deliver gene therapy, and we want to develop one that's specific to FOXP1. So we have a proposal to do that from leading experts in the field, but it's going to cost $150,000 a year for 2 years, and we don't have that money right now. And they're going to move on to something else if we don't fund them. So we need to raise that money, and we need to raise it very soon. Okay, so that's why this star is for funding funders. All right, let me talk about ASO upregulation because we're investing a lot of money into this, and this is something that was mentioned yesterday. I got some questions about what is it. So, so this works on the RNA level, which is has a plus and a minus. So what it means is that you get a treatment because RNA is degraded in the body. You have to get this treatment, say, every 3 months. Okay, so it'd be a regular treatment. It's not a one-time thing like a gene therapy that that Cheryl spoke about earlier. This is something you get every 3 months, and it would be with this lumbar intrathecal injection that you just talked about, kind of similar to it, getting an epidural. Sorry, I'm going to go over. Okay. So let me explain what an ASO is, or an ASO upregulation. This is not a standard ASO. This is what's called an ASO upregulation. So this is, for the experts, this is a vast simplification of what's really going on, but I think it'll be helpful. So, and this is, applies to basically all of our kids. So all of our kids pretty much have one copy. So everyone has 2 copies of, each chromosome other than the sex chromosomes. And so on the 3rd chromosome is the FOXP1 gene. And this is mistake because I stole this slide from another presentation on another gene. And oops. And the FOXP1 is actually up here. It's not down here. But anyways, the point is that one of the two FOXP1 genes has a mutation in it. There's some mistake, okay, for each of our kids. And typically this is not inherited. This is just what's called de novo mutation, which means it was a new mutation that came, when the child was conceived, basically. Okay. And so normal FOXP1, each of these genes gives rise, so the DNA gives rise to RNA through a process called transcription, and then the RNA encodes the production of a protein through a process called translation. And so a normal FOXP1 gene gives rise to, or maybe normal is maybe a little bit judgmental. I think it's called wild type. The slide should say, typical gives rise to a typical functional FOXP1 protein, whereas a mutated gene gives rise either to a protein. So this is again a simplification. Usually not no protein, but maybe a truncated protein that then gets degraded, or a mutant protein, so a protein that has some aspect to it that it's not the same. There's basically an amino acid sequence that encodes this protein that is different in this protein, and so it's not functioning the same way. Long story short, usually both copies are producing a protein, and now only one copy is producing a protein. So roughly speaking, you have half as much functional protein as you should. So that's the takeaway from this slide without making it too complicated. For all our kids, they need 2 of these proteins and they're only getting 1. Okay? Now, there can be more complicated than that. This other protein could be doing something, but I don't want to get into those details. Let's just focus on the fact that they're getting half as much protein as they need. Okay, so what is an ASO upregulation? ASO upregulation, it doesn't actually try to fix the mutated protein. It does something different. What it does is it goes into the healthy copy of the gene and it puts in something called an ASO, which basically binds to some regulatory elements on the RNA that's leading to the translation of this protein to try to get it to produce more than it normally does. So it's going to the healthy copy of the gene and saying, hey, can you produce more for us since you're producing good protein? And so we're kind of compensating for the fact that the mutated protein is not producing good protein. Okay, that's what an ASO upregulation does. You get this increased protein output. Okay. All right, so I'm way over time. Let me just say, we have lots of projects for future research investment that we like. So I'll just say about the ASO, we're investing in that. So we're doing the research to try to establish the science behind that now. We're just starting. We just approved that funding. There's lots of research that we need to move forward with, and I won't go because I don't have time. I'll just say one kind of what I think is low-hanging fruit that we should have probably done even before that, again, we would love to do this if we had more funding, is there's a screen by a company called Unravel Biosciences, which would take 20 samples from patients in our community and try to understand if there's any drugs that exist out there now that can help our kids. This is called drug repurposing, drugs that are approved for other conditions that they can determine by understanding the genetics and running their proprietary screen that it will help. All right, so now let me end with some brass tacks. So I'll be more blunt, less bashful than Carl was about asking for money. Yeah, okay. Oh, okay, sorry. So in 2024, the funding that we had was primarily founded by 4 families. So, this family one is my family. So 60% came from one family. This is not sustainable. So we need more donors and we need larger donors. So I'm really asking, I hope I've done somewhat of a good job saying that, we're good stewards of your donations. We're really funding research effectively under the guidance of our SAB. And I really think we can do this in the years ahead. And I think you can probably feel that things have moved forward a lot in the last year or two in this. And thank you very much. Let's do this.